Assalamualaikum and hi guys. So now we go to the final chapter in our syllabus, which is chapter 5, the introduction to biochemical reaction system. Okay, so this chapter or this topic is actually chapter 7 in your Fogler book. So you may refer to chapter 7. But before we go further, I would like to tell you that this chapter in Fogler, chapter 7, will not fully covered. Okay, so we only focus on certain part in chapter 7. So I'm going to teach you only on the inhibition of enzyme for this chapter. Okay, right. Okay, so if you look at the aim of this topic is to provide an introductory overview of how biological system can be used by chemical engineers. So we only then on the surface part of this biochemical reaction system. Okay, right. So these are the learning outcomes for this topic. You need to demonstrate and understand of the kinetics of enzyme catalyzed reaction. Okay, explain the lock and key and induced fit model, demonstrate an understanding of the mechanism of enzyme action, and then understand how to use Michaelis-Menten equation and to apply this equation to the analysis of enzyme kinetics. So if you look into the learning outcome, it's all about enzyme catalyzed reaction, right? Okay. So, this is the overview, okay? So, it's related, biochemical engineering is related with design, development, implementation, operation of engineering process. So, these are the related disciplinary, okay, to understand on the biochemical engineering, okay? So, what is bioprocess? So, bioprocess operation is the use of microbial, animal, plant cells, and component of cells such as enzyme to manufacture new products and destroy harmful waste, right? So these are the applications for the biochemical reaction or industry. So we can use it in chemical industry, food, okay, and medicine. So I think you can go through this in your textbook, right? So these are the introduction to the bioprocess. Okay, as you've seen in this figure, this is actually a bioreactor. So in order for you to optimize the performance of the bioreactor, these are the factors that need to be considered. Okay, the concentration and time, temperature, pH, the availability of nutrients that you're giving into the bioreactor, okay, the source of carbon, source of water, electron acceptors. So these are the factors that will affect the performance of the bioreactor. So you see there's a steam coming in to sterilize the bioreactor, right? And then you have pH controller, cooling jacket so that the temperature will not exceed the temperature of the microbe can live in, such as normally it will optimize until 40 degree, I think the maximum, okay, but there's certain microbe that can resist high temperature, okay. So the next one, okay, these are the important parts that we need to learn. So enzyme catalyzed reaction. So as you know, enzyme is also one example of catalyst, okay. So it is a biocatalyst, okay, in other words, right? So what is enzyme? They are produced only by living organism, okay? So, okay, produced by living organism, okay? And commercial enzymes are generally produced by bacteria. Most of enzymes are high molecular weight, okay, because it is protein, okay? And then, it acts on substrate, okay. Substrate is the reactant of the molecule, okay, like the one you learn in producing in chemical reaction. So you have substrate, right, or you have a starting material A. So like A producing B, so this is your initial or your reactant, okay. So in this case, reactant is actually the substrate. Okay, right. Transform it chemically at a rate, accelerated rate, usually 10 to the power of 3 to 10 to the power of 17 times faster than the uncatalyzed rate. Enzymes are specific. Okay, enzymes are specific. This is very important. One enzyme can usually catalyze one type of reaction. Okay, so the next one we're going to learn on how enzyme, okay, the reaction steps that use enzyme, the reaction step involved in the using of enzyme. So enzyme are present in small, okay, small quantities. Okay, sorry. Right, let me use my pen. Okay, small quantities. Okay, and then are not consumed during. This is important. Not consumed. Okay, it's simply same like catalyst. Okay, enzyme is actually equals to your biocatalyst. Okay. Okay. So uncatalyzed reaction. This is substrate. This is product. So this is your enzyme. Okay. So it will make enzymes substrate bond, and then it will release the product, right? So catalytic activity is revealed by studying enzyme kinetic. Okay. So this is activation energy. I know that you have seen this before in chapter 1 for your catalyst reaction. Okay. So you see here it's the same thing, enzyme and catalyst. So the blue one is with enzyme. The red one is without enzyme. Okay. So you can see that with enzyme the activation energy is lower down. Okay. So enhancement in reaction rates can be enormous, right? Okay, the next one, enzyme substrate complex. Okay, so you know that substrate will bind into specific enzyme to form an enzyme substrate complex like this one. Okay, lock and key model. It has two, lock and key model or induced fit model. So this is what, okay, so this is the part that we call enzyme and enzyme substrate complex. Okay, right. So this is how the process or the mechanism of enzyme reaction that catalyzed by enzyme. So the first step, okay, if you look here, the first step, enzyme, this is your enzyme, the purple, and then substrate, which is in this case sucrose. The enzyme is sucrase. So enzyme will, the substrate will go to the enzyme and form enzyme substrate complex. Okay, and then it will convert to product. Okay, so this is product enzyme complex. Okay, and then it produces this product that consists of glucose and fructose, and the enzyme will go back to the initial condition. So it is like the concept of catalyst, where it will not change throughout the reaction. Okay, it's ideally. Okay, right, this is enzyme kinetic. Okay, so if you have let's say you are running an enzymatic reaction. Okay, so it's enzyme plus substrate. It will produce enzyme substrate and then product and enzyme. So what you will record, this is substrate concentration, this is your reaction velocity, or what we call it your reaction rate. Okay, so it increases when your substrate concentration increases. It's just the same like your chemical reaction, right? Until it reaches a certain maximal, a certain condition of substrate where it reaches the maximum velocity, or where it reaches the maximum reaction rate. Okay, so we will use this mechanism-maintain kinetics in order for us to develop reaction rate for the to describe the reaction rate of enzymatic reaction. So the enzymatic reaction can be described by mechanism-maintain equation, which is described by this equation Right? If you look here, ds over dt is equals to negative rs. So last time we have learned, right? Remember dca over dt is equals to negative ra equals to kca. Similar by this is the change of your substrate. Okay, this is your the reaction rate due to the enzymatic reaction. So this is the part where is the mechanism maintain equation. So S is the concentration of substrate. Vmax is the maximum rate of reaction. Remember just now this graph. This is your Vmax. Okay, this is your Vmax. Okay, right. So Km is the Michaelis constant for the substrate. So in your case, you will need to determine the Vmax and Km, right? So this is when you plot the graph of Rs versus S. This is your or in the earlier slide it was your maximum the velocity of your reaction. Okay, so when you plot this one, so you can determine your Km when it is equals to half Vmax. So this is your Vmax when your substrate is achieving it's not changed throughout the reaction is not changing when your substrate is increasing. Okay, so this is your Vmax. So you will need to determine what is your Km. Km is half of Vmax. So this is your Km. Okay, so besides then determining the Vmax and Km from the graph, okay, we can convert it into a linear equation like the one that used in your textbook. Okay, so this is okay, this one is just some elaboration on the Michaelis-Menten kinetics. Okay, so in order for you to determine easier the Vmax and Km, we can convert. Okay, we can convert the Michaelis-Menten equation, this equation, right? This equation into a linear equation. Okay, so you linearize this equation. So when you linearize this equation, it will be what we call it the one equation in your textbook, page 403, okay, and 402. Okay, it is what we call it Lin-Weaver-Burk plot. Okay, so let's have a look on here. Okay, this is the equation that I've been telling you just now, the Lin-Weaver-Burk. Okay, if you scroll into this page, you will see, okay, right, where is it? It's not stated here, but okay. So this is what we call it, the Lin-Weaver-Burk plot. Okay, so you may use this equation, right? So it is very important one. Okay, where you need to determine your Km Vmax. Okay, so this part is actually your C. Okay, your C, and this part is actually your slope, Km over Vmax. Okay, so let's have a look on the example. Okay, so initially they will give this data. Okay, C urea and the reaction rate of urea. So following the Michaelis-Menten, you can plot this graph. But for you to for easier to determine your Km and Vm, you transform it into a linear graph, which you use the Lin-Weaver-Burk plot. So just convert the equation into Lin-Weaver-Burk. So that's why you are plotting back 1 over the concentration of urea and 1 over Rs. So it's Rs 1 over Rs versus 1 over Cs. Then you will get a straight line here. So you can determine, okay, so this is actually your C, which is your 1 over Vmax. And this is your slope, which is Km over Vmax. Then you will get your Km by replacing the Vmax that you obtained earlier. Right, so I stop here. We'll continue in the next video to tell you, to show you on the inhibition factor on the enzymatic reaction using the Lin-Moberg plot. Bye!